Today's meeting is going to be recorded, and we will post that at the NISS website. I don't have an exact address yet, but if you look on the NISS website, after you give us a little time to process the recording, we plan to post it there. The basic topic for today is FDA draft guidance on multiple endpoints in clinical trials, certainly a topic that everybody in pharma is very interested in. Okay, so what is the agenda for today? First, we're going to have Ray Bain from Merck answer the question for us, what is NISS? Then Alex Dmitrienko is going to give a presentation on multiplicity guidelines. Frank Bretz is then going to talk on the implementation of the FDA guidance. And then we're going to have a panel discussion. The discussion will be moderated by Walt Alphin. And we have distinguished panelist Lisa Lavange from FDA. And the other panelists will then also add their comments. And hopefully we'll have a nice, lively discussion. So with that, I'd like to pass it to Ray to talk about who NISS is and why they're an important organization.
Thank you, Dan. So as indicated, I will do a little presentation here on what is NISS. Obviously, Dan has already indicated that they are a co-host of this event today. So the National Institute of Statistical Sciences, or NISS, was formed in 1990 with the vision to identify and foster cross-disciplinary research involving the statistical sciences. Since then, cross-sector research and addressing critical interfaces between industry, government, and academia has become an increasingly central role to the NISS portfolio. NISS's mission is dedicated to serving both the U.S. and international statistics community by engaging the NISS community membership participation in applied research driven by problems facing government and industry. So what is NISS today? I will highlight 6 characteristics of NISS. First characteristic is that NISS is a national research institute, and they have a 27-year record that advises and collaborates on scientific research and public policy. NISS works on projects where information and quantitative analyses are the keys to the solution. They have the capability to identify experts in the statistical community to address specific research topics. Characteristic number 2, NISS is a 501c3 nonprofit organization with governance by a board of trustees from academia, industry, and government, totaling about 20 members. The structure of NISS includes internal staff consisting of experienced senior statisticians, postdoctoral fellows, and technical administrative staff. The third characteristic is NISS is a postdoc program, and it's one of the oldest, largest, and most successful postdoctoral fellowship programs in statistics. The program has provided training of more than 80 early career statisticians. The postdoc program is organized to achieve high-quality collaborations by pairing experienced NISS senior leadership with NISS postdoctoral fellows. These NISS postdoctoral fellows have NISS technical mentors and also practical mentors from a substantive field who work together to solve the scientific and public health, public policy problems. In so doing, the postdoc fellow gains technical research experience as well as developing the soft skills necessary for scientific collaboration. The fourth characteristic of NISS is that it is an independent research organization, and they have an established base of grants and projects currently funded by federal agencies. Examples of these federal agencies include the National Center for Education Statistics, the National Agricultural Statistics Services, the U.S. Environmental Protection Agency, the National Cancer Institute, and the U.S. Department of Transportation. The fifth characteristic is NISS is a co-sponsor and coordinator of conferences, workshops, and expert panels for the statistical science community. For example, upcoming NISS conference workshops in the second half of 2017, in addition to this particular meeting, is they are co-sponsoring with the Washington Statistical Society a workshop on inference for non-probability samples. They're also sponsoring a workshop around R and Spark, which are tools for data science workflows. There's one of the workshops on the East Coast, one on the West Coast. And they're also sponsoring Design and Analysis Experiment Conference. And the sixth and last characteristic that I want to highlight for NISS is that they are a communication enabler. And this is particularly for the statistical science community through the NISS Affiliates Program. The Affiliates Program consists of academic, government, and industry members. The program enables research discussions, information dissemination, and networking with a goal to use statistical sciences to solve problems facing industry and government. Benefits of participating in the NISS Affiliate Program include a priority for NISS program registration. So for NISS-sponsored conferences and workshops, you get priority registration for those, and also you get reduced fees for the affiliate members to participate in those programs. Second is a priority for NISS special topics workshops. So the special topics are determined by the affiliate members. They are organized by NISS and then led by the affiliates. And a typical format of a special topics workshop is a small group of senior research convened by NISS to interrogate the role of statistics in a new or growing area of interest. And the last benefit for participating in the affiliates program is the annual affiliates meeting, which is held at professional society conferences or NISS-hosted or co-sponsored conferences. And the program is planned by the affiliates committee. So those are my 6 highlights of the NISS organization to give you a better idea of their role and responsibility going on. And for additional information or more information on the 6 highlights that I've provided, you can see the NISS website at www.niss.org. And with that, I will hand it over to the first speaker.
Well, actually, I'll introduce I apologize, Ray. I did not introduce you first, so I should have started that. Okay. Ray, of course, is the head of Merck's Biostatistics and Research Decision Sciences organization, sometimes called BARDS, and has been the head since 2001. That's a global organization which has biostatistics and programming responsibilities across practically all the phases of drug development at Merck. Since 2015, Ray has also served as vice chair of the Board of Trustees at NISS, which of course is the National Institute of Statistical Sciences. So thank you very much, Ray, for filling us in on that. Now we're going to move to Alex Dmitrienko's presentation. Let me pass him the ball so he can share his desktop. And while he's doing that, I will introduce him. Alex is founder and president of Mediana Incorporated. He has over 20 years of clinical trial experience and has been actively involved in biostatistical research with an emphasis on multiplicity issues in clinical trials, subgroup analysis, innovative trial designs, and clinical trial optimization. He has co-authored or authored a number of books, including 2 from SAS Press, Analysis of Clinical Trials Using SAS and Pharmaceutical Statistics Using SAS, and 2 from Chapman and Hall, Multiple Testing Problems in Pharmaceutical Statistics and Clinical Trial Optimization Using R. He has served as an associate editor for 3 of my favorite journals, the American Statistician, Biometrics, Statistics and Medicine, and as a fellow of the American Statistical Association. And with that, I hand it to you, Alex, and multiplicity guidelines.
Great, Dan. Thank you so much for this introduction, and thank you for inviting me to contribute to this meetup. This is the first time I'm contributing to a NISS Merck-sponsored meetup, and I'm very happy to be here. I hope everybody can hear me well. And right now, it will be my pleasure to provide a high-level review of regulatory considerations and guidelines related to addressing multiplicity in clinical trials. So we're going to begin with a list of applicable guidelines. And those would be, of course, the FDA guideline or guidance, draft guidance on multiplicity issues in clinical trials that was released in January 2017. I would also like to mention two relevant guidelines that were published by the European Medicines Agency. That is the draft guideline on multiplicity issues in clinical trials that was released earlier this year. And this was, in fact, a revision of points to consider on multiplicity issues in clinical trials that was published in 2002, which was 15 years ago. And that really tells you that time flies. And when we begin with a high-level comparison of the general philosophical approaches that are presented in those two guidance documents or guidelines, there was an important discussion that took place at a recent conference, the CN-ISBS conference that was held in Vienna, Austria, a couple of weeks ago. And it was recognized at that conference that the really fundamental difference between the way those two guidelines or guidance documents approach the general topic of multiplicity is that the FDA guideline provides information on key principles as well as underlying statistical methodology, and by contrast, the EMA guideline focuses mostly on general principles. As a result, this guideline document is much shorter. In this presentation, in the interest of time, mostly I'm going to focus on the key points from the FDA guidance document. And I guess the reason for that is that we all recognize that the impact of this guidance document will, of course, reach beyond the U.S. borders, because the opinions and guidance of the U.S. Food and Drug Administration are very influential with clinical trial sponsors and regulatory bodies around the world. I would like to begin by saying that in this draft guidance, the FDA provided its current thinking on the multiplicity problem imposed by multiple endpoints in the analysis and interpretation of clinical trial results. And when we begin with this topic of multiple endpoints, the guidance offers a very comprehensive review of available multiple testing methods. It provides numerous practical considerations and even certain technical details. So to facilitate the review of this guidance document in under 15 minutes, that's how much time we have right now, I've decided to go ahead and select a few important topics, and I think that those topics will help set the stage for the discussion that we're going to have after these presentations. But let me first of all address this very important point that has come up multiple times whenever we started talking about the FDA guidance's scope. Formally, this guidance document, of course, focuses on multiplicity induced by multiple endpoints, but I would like to use this quote from section 2 of this guidance document. And this quote emphasizes the important fact that the issues, multiplicity issues that are relevant in the analysis of multiple endpoints are actually broadly applicable to a large number of other settings that arise in clinical trials that employ multiple doses, multiple time points, or study population subgroups. And therefore, this document actually addresses a very broad range of multiplicity problems that we encounter in clinical trials. Now, if we begin looking at those key topics I've decided to select, I have a few slides here, and I'm going to go through those key topics one at a time. We're going to begin with the type 1 error rate control. This FDA guidance document provides a very clear definition of what we mean by type 1 error rate control in confirmatory trials. It gives a formal mathematical definition of family-wise error rate in the strong sense, which is the current approach used across all of confirmatory trials. And this guidance document also clarifies that this definition of error rate control must be applied at a trial level. And this needs to be contrasted with other proposals that we may have seen in the literature, that perhaps error rate control must maybe performed at a program level, or perhaps we could even consider control over multiple regulatory regions. This is not how we are supposed to define type 1 error rate control in confirmatory trials. I've also noticed that this guidance document provides a very detailed coverage of different settings when it comes to multiplicity in clinical trials. It begins, of course, with a traditional setting, which is characterized by the fact that we have a single component of multiplicity or a single source of multiplicity, for example, multiple doses compared to a control. It also spends a good amount of time discussing the more relevant, I would argue, advanced setting, which is something that we see in most modern clinical trials, where we have to deal with several sources of multiplicity simultaneously. And what I mean by this is that we may have several components, and each one of them in and of itself induces multiplicity, and all of them are analyzed at the same time within a clinical trial that includes multiple dose control comparisons, perhaps in several patient populations, or perhaps for several clinical endpoints. So in those cases, we'll have two sources of multiplicity. The guidance document also defines commonly used success criteria or win criteria that we see in confirmatory trials. For example, at least one clinical objective is met, or all relevant clinical objectives are met. And as a result, it also introduces appropriate multiplicity adjustments that are aligned with the criterion used in the trial. The second set of topics here is related to the analysis of hierarchically ordered endpoints, or more generally, hierarchically ordered clinical objectives. And this is a topic I would like to spend a bit more time on, because when we take a closer look at this topic, we realize that the guidance document provides very detailed information on secondary endpoints that play a key role in informing risk-benefit assessments and providing useful information for the prescribing physicians and patients ultimately. And the guideline clarifies the role of secondary endpoints with the dichotomy of secondary endpoints, I should say key secondary endpoints and exploratory endpoints. And the difference here, of course, is that inferential statements could be made for the predefined secondary endpoints, which means p-values and or confidence intervals could be presented in the product label, but inferential statements are not permitted for exploratory endpoints because they're not covered by alpha control. Another point I would like to highlight here is that this guidance document does a very good job of introducing commonly used multiple testing procedures. And I cannot overemphasize the importance of this topic, because it actually goes back to my quick comparison of the philosophical approaches in the FDA and EMA guidance documents, because there are very useful specific recommendations presented in this guidance document, which is something not explicitly covered in the EMA guidance. And this was, in fact, the feedback that was shared at the recent conference in Vienna. The general principles are, of course, very helpful, but even high-level advice or even the references would have been greatly appreciated. And unless one is very knowledgeable in multiple comparisons, it may be quite challenging sometimes to interpret certain recommendations presented in the EMA guideline. And with the FDA guidance, everything is quite transparent, and as I said, all popular multiple testing procedures are presented in this guidance document, and this is where I think the FDA guideline really shines. Continuing to a number of more technical topics here, I would like to point out the fact that the FDA guidance recognizes fundamental facts such as the importance of an analytical approach to deriving valid multiplicity adjustments. This needs to be contrasted with simulation-based approaches that are not appropriate, but surprisingly, I still see multiple attempts to justify alpha control by using simulations for some complex multiplicity adjustments, and that's why I'm very glad that this statement is made in the FDA guidance. For power evaluations, the guidance clarifies that simulation-based evaluations to support power and sample size calculations are recommended. In fact, that naturally leads to a very important topic of clinical trial optimization, because by using appropriate simulation-based approaches, a clinical trial sponsor can easily evaluate the performance of multiple candidate multiplicity adjustments and select the one that is most clinically relevant and provides the best performance for each particular setting, for example, each particular trial. I would also like to very briefly mention a few topics that are not covered in this FDA guidance. This will not be a criticism of the guideline, but simply to recognize the fact that certain topics are beyond the scope. Of course, it's impossible to cover everything. This is not, of course, the Internal Revenue Code. So I'd like to say that multiplicity issues in early stage trials are not explicitly addressed. This also applies to multiplicity in exploratory settings, and that's something that is relevant in the analysis of safety endpoints, for example, or in post hoc subgroup analysis. But at the same time, there are multiple publications that address those multiplicity issues. And when it comes to multiplicity issues arising in adaptive trials, there's actually, as we know, a separate FDA guidance that covers this topic. And now that we're almost done with the discussion of topics that are presented in the FDA guidance, I would like to very quickly mention that, of course, not all of the multiplicity-related topics are explicitly addressed in this guidance, but the good news here is that there's a number of recent review papers and tutorials that I would like to mention a few of them on this slide, and they will, of course, provide more information about some of the technical details. And now, very briefly, we know that the release of the FDA's and as well as the EMA's guidelines encouraged all of us in the industry to begin discussing the role of multiplicity in clinical trials. And here I would like to mention very briefly multiple communication channels and forums for discussing multiplicity-related issues. Would like to mention, first of all, that Statistics in Medicine is going to publish a number of commentaries on the FDA and EMA multiplicity guidelines, hopefully at the end of this year. There will be a special issue on multiplicity issues in clinical trials that will be published in early 2018 by Journal of Biopharmaceutical Statistics. I'm happy to report that there will be contributions, mostly technical papers, from statisticians who represent regulatory agencies, industry, and academia, different regions. If you have a chance, I would like to invite you to attend a session on multiplicity issues in clinical drug development that will be held on Tuesday, September 26th at the Regulatory Industry Statistics Workshop in DC. In just a couple of weeks, you'll see that the speakers will be Dr. Lavange and myself. At the very last moment, Professor D'Agostino told us he would not be able to present at this session, but I still think this would be a very important discussion. And one last point is that the ASA Biopharmaceutical section has been working hard to set up an online training program, and I would like to mention that this program has attracted a good amount of attention across the pharmaceutical industry, and there's a full-day course on key multiplicity issues in clinical trials that I would like to invite you to take advantage of. It's a very convenient and inexpensive option, which is supported by the biopharmaceutical section. So to summarize, this guidance document, I believe, provides a solid introductory tutorial on methods for maintaining strong control in confirmatory clinical trials. And I believe that the information and the recommendations presented in the FDA guideline will encourage drug developers to learn more about novel approaches and also judiciously apply them in confirmatory clinical trials. So I will stop at this point. I also have a few slides with references that you may find useful, but at this point, I would like to thank all of you and would like to turn this over to Frank.
Thank you very much, Alex. That was extremely illuminating. You hit all the major points. So I'm going to pass the ball to Frank. And while I'm doing that, let me introduce him. Frank Bretz is joining us from across the ocean today, so thank you, Frank, for that. He's been at Novartis since 2004, where he's currently the global head of the Statistical Methodology and Consulting Group. He has supported methodological development in various areas of drug development, including dose finding, multiple comparisons, and adaptive designs. So, Frank, the floor is yours.
Okay, thanks, Dan, and thank you for the kind invitation to present here today. And after this nice review by Alex on the FDA guidance and building upon his elaborations, I have been asked to provide a few comments on the implementation of the FDA guidance. I guess you can see my slides as I'm flipping through them. So, one of the implementations is, at least in my organization and as much experience as I have, is that we like to use the FDA guideline as a rational clinical endpoint classification when talking to clinical teams. What I mean by this is that we interpret the guidance by having a very clear discussion what is primary, what is secondary, and what is of exploratory interest. So you see here in a tabular format a little bit how we internally characterize and how we interpret the FDA guidance, primary endpoints being those that are necessary or sufficient to establish efficacy and meaning trial success. Secondary endpoints are additional meaningful outcomes that represent alternative facets of the disease, not just minor variations on other endpoints. And exploratory endpoints could be hypothesis generating or variations on primary, secondary endpoints. And internally, we also tend to link these characterizations with implications on approval and product labels, and also the amount of the type 1 error rate control that is necessary, as you see it here on this slide. So moving on, the FDA guideline, however, also helps in terms of this, because it's so scientific and methodologically oriented, as Alex already pointed out previously, it has a very complete description of methodological work. It also offers room for future, research and methodological work. And so I just have one little example that we came across more recently, where we are planning 2 identical studies with the same 3 clinical outcomes, which are of hierarchical importance. So of primary importance, you would like to see a reduction in a short-term symptom used conventionally for regulatory purposes, whether the new drug is effective. But then the second endpoint is a reduction of a long-term clinical endpoint, which is typically underpowered when you would plan for a single study, and then you might also be interested in a patient's quality of life score. And the question is how you come up with a suitable multiple test procedure that meets the practical constraints and specifically sample size limitations that could arise from your secondary endpoint, which is typically underpowered indeed if you would plan for a single study. And so there are other practical considerations as well, but you can use the current guideline and start from there having a good discussions, regulatory discussions. And one possibility is, and I'm showing that just very briefly on the next slide, and in the interest of time, I'm not going to any details, but you see that we came up with an example strategy that uses a hierarchical test strategy where you test through the 3 variables in hierarchy according to clinical relevance with a separate testing possibility of your long-term symptom variable 2, which then uses the pooled data across both studies. there needs to be some additional thinking coming in, how you would allocate your type 1 error rate or your alpha, your significance levels, because you're pooling data across studies, but you still want to control the type 1 error rate within a study. So there are some additional considerations, and again, in the interest of time, I'm not going to any technical details here, but this is just to illustrate how the guidance, the current FDA guidance, helps us to move on into, and addressing new problems that are currently not being addressed in the FDA guidance. And as Alex pointed out before, you can't expect all problems and types of multiplicity issues being covered by a guidance. So there are some key points that I tend to tell my teams as well, which probably go into like good practices of drug development and designing trials, but which are also related, or in particularly related, to multiplicity and multiple endpoints in our phase 3 trials. So first, we feel it is important to get agreement within clinical teams, that includes statisticians, clinicians, marketing, and other line functions, on which endpoints are critically important, which are clinically relevant to gain approval and are to be included in the label, as opposed to those which are less important and should be considered more exploratory. And having this guidance that helps us in our internal discussions in getting clearer and more transparent discussions on the value of various endpoints. Secondary endpoints are now those that previously were often labeled as key secondary endpoints, whereas regular secondary endpoints in previous times are now possibly considered as exploratory. Also, we should avoid clinical trials with a rather large number of hypotheses and avoid trying to salvage a study with convincing secondary endpoints. And that is another key point that we take out from this guidance as we implement it. Finally, it's good to see that a number of graphical and great gatekeeping procedures for type 1 error rate control are accepted by regulatory agencies and are possibly seen as a mean to facilitate the discussions within clinical teams. That also helps us to compare the operating characteristics of competing decision strategies by using different candidate multiple test procedures using tailored clinical scenario evaluations. So that is an important aspect and should be foreseen to be used much more in our day-to-day practice when designing our big phase 3 trials, that we do understand the operating characteristics and the performances, so to speak. In the end, even the best methodology cannot make up for a bad choice of the endpoints or of the multiple test strategies if these are not aligned with the business decisions related to market approval and product labeling. So having this guidance helps us to having an upfront discussions about the importance of the various endpoints and how we want to test them in the right sequence, so to speak. With that, I guess I'm handing over again to Dan.
Thank you so much, Frank. That was a great summary and really some excellent points there. So thank you. Okay. So now, again, it's sort of keeping along the lines of a meetup where you have discussion. The idea now is to discuss these points. And so to do that, it's working here. Trying to get. Let me introduce them, and I'll put the picture up when I get to it. So to moderate the discussion, we're lucky to have Walt Offen, who's currently Distinguished Research Fellow and is the head of Statistical Innovation in Safety Statistics and Non-Clinical Statistics at AbbVie. He joined AbbVie in 2012 after a 31-year at Lilly. He has had a long career of interest in research in multiplicity topics. So thanks for joining us, Walt. And let me introduce Lisa while I'm at it, and then I'll turn it over to you to moderate the discussion. We also are lucky to have Lisa Lavange, who's Director of the Office of Biostatistics in the Center for Drug Evaluation and Research at the US FDA. As a director, she oversees approximately 225 statistical reviewers and staff members involved in the development and application of statistical methodology for drug regulation. So thank you very much for joining us, Lisa. And with that, I turn it over to you, Walt.
Great. Thank you, Dan. First thing I want to say is that any of the attendees, if you have questions, please type them into the chat box there, and we'll try to address as many as we can. I have about 6 or 7 questions that I created last night, actually, and so a heads up to the panelists, I have not shared them with any of them, so they'll all be caught off guard a little bit. Where I want to start is I want to give Lisa an opportunity to either comment on either of the 2 presentations or ask questions of those presenters, or if there's one kind of burning issue, Lisa, on your mind of things that you see that are not adequately or appropriately controlling for type 1 error that you would like to point out, give you the first shot, Lisa.
Great, thanks. So I would just like to thank both of the speakers for really nice talks, and also to Ray and Dan for organizing this webinar. We are at FDA very excited to get this guidance out there published. It was actually a commitment under our 4th passage of the Prescription Drug User Fee Act, which is about 8 years ago now. So we were happy to publish it last year, or I guess in January of this year. And we did receive a number of comments, and we've poured through the comments left on the public docket, and we're now in the process of finalizing the guidance, and we'll hopefully get a final guidance published in the not too distant future. Both of our speakers touched on the key issues. Alex talked about the in-depth explanation of some of the more complicated methods, particularly the passing of alpha levels from one family to another, some of the graphical techniques that are in the appendix of the document, and so forth. And I'll have to just mention Alex and Frank both were 2 of the people who published a lot of these methods to begin with. So they're both expert in the methods that we are showcasing in the document. We interestingly got comments from the public. Half of the comments said, why do you need to put this tutorial inside your guidance, because by now it's already in the literature, whereas the other half said, oh, we love the tutorial being in the guidance. So we'll have to make a decision about whether we hold on to that tutorial in the final guidance or just reference it with the now published literature. But I do agree with Alex. I think it's nice to have a nice description of the methods all in one place. Frank, in talking about how the guidance is practically applied, I think anything that helps guide a clinical statistical discussion about which endpoints are important can do nothing but help drug development. I think that there are so many things that can go wrong by focusing on the wrong endpoints or having too many endpoints, or so forth and so on. And so I really like that message. I also like Frank's proposal about the hierarchy of the symptom and then the long-term endpoint that needed pooling across trials. We got comments about the fact that we don't give any guidance in the document in its current form on what to do if you do need to pool trials to get enough power for an endpoint that might be mortality or severe morbidity that is tough to find in a study that's otherwise more than adequately powered for a symptom or a shorter-term endpoint. And so that is something I think we need to address in the final document, and it has in fact been discussed at a number of advisory committee meetings where it's clear that the pooled data can inform a decision, but isn't really accounted for in any of the alpha spending functions that we talk about in the guidance. So that's something Frank, I think, hit very well that we'll need to address. And the only other thing I'll mention quickly is that we got some questions in the guidance. Why not talk about multiplicity in general? Multiple endpoints is only one area of concern for multiplicity in regulatory actions and in trials designed to support regulatory actions. We've got other sources like multiple doses, multiple analyses of the primary endpoint, multiple analysis populations, and then, of course, multiple subgroups. And you know, we got questions about whether we intend to expand to a full-blown multiplicity guidance. We do not right now. The multiple endpoints we felt was very important, but we do acknowledge a lot of the methodology might be applicable to these other sources of multiplicity as well. And then we got questions about whether strong control was always needed across secondary families, and in particular, what about safety endpoints that are secondary, not primary but secondary, and how do those endpoints fit in? A couple of other comments, a couple of other areas where we got lots of comments were about rare diseases, and Frank mentioned areas for future research. To me, the heterogeneity of rare diseases, which are also tough to study in anything other than a very small and usually underpowered clinical trial, is a real problem. And there have been some public workshops recently to talk about individualized endpoints for these very heterogeneous but severe rare diseases. Of course, the guidance could not go into anything in depth there, but I think that problem, I think a related problem about multi-component endpoints, particularly patient-reported outcomes that might have an overall scale and subscales, those are also not covered and might be areas where we need more research and potentially future guidance coming from FDA. So I'll stop with that and just thank you again for the opportunity here, Walt.
Great. Thank you very much, Lissa. Let me start. I'm going to go to a question that was in the chat, but before I go there, I want to ask a quick question to Frank Bretz, and it's slide 12. I don't know, Dan, if it's easy to bring it back up or not, but if not, Lissa referred to it, and it's where you have two studies and you have a graphical method where you split out some of the alpha to test a hypothesis that pulls across the two studies. My question is, what would you and the other panelists recommend that alpha be? I mean, normally study 1 would have an alpha 0.05, study 2 would have an alpha 0.05. You certainly wouldn't allow something more than 0.05 to go to H2, right? Do you have any comments, any of you?
Yeah, maybe I could just give it a start. So you would firstly, I mean, H2 tilde, that is like how we call this pooled hypothesis, so to speak, or where the hypothesis for the secondary endpoint based on the pooled data. Now, the question is, you know, how do you, after having established significance for the two primary hypotheses, H1 and H1 prime, from the two studies on the very same endpoint, you know, the question is, how do you now allocate alpha for the remaining 3 hypotheses? And you know, one possibility would be a Bonferroni split across H3, H3 prime, and this H2 tilde. Now, then you can have considerations as follows, that H3 and H3 prime are stochastically independent. So the type 1 error to reject both at level alpha becomes at most alpha squared. So on the other hand, if only the pooled analysis were done, its significance level would be alpha. So that gives one rationale, for example, of using alpha minus alpha squared for H2 tilde, which then would control the family-wide error rate for each study at level alpha. And so there are some ideas along these lines, and probably that was a little bit quick now, but there are some, you know, I think starting this Bonferroni split with a wise considerations of what you want to control within a study and what you want to control overall, I think that these are some of the guiding principles, I would think.
Okay, thank you, Frank. Alex or Lissa, any other comments to that?
This is Alex. If I might go ahead and maybe make a quick comment related to this. And maybe if I could just step back a little bit to just talk about some of the discussions that we had as early as the multiple comparison conference that was held in Washington, D.C. back in 2011. There was a panel discussion that was included multiple experts from academia and in industry. We even published a white paper in the Biomedical Journal to discuss the advantages of using pooled analysis of secondary endpoints in general, because we see a lot that secondary endpoints are underpowered, and with the pooled analysis we're going to end up with improved power. And we also see that it's rather common to see inconsistent results across different trials within the development program when those secondary analyses are performed within each individual study, even with a proper alpha adjustment, which leads to some frustrating results. So in this case, if I may come up with a more ambitious proposal or a more ambitious question, and that question would be, why not simply pull the analysis, pull the data, from those two trials for that variable 3 as well. That would have a number of advantages that I've just outlined. It will, in addition, simplify the process of applying a multiplicity adjustment, because then variable 2 and variable 3 will be in the same boat.
Okay. Thank you, Alex. Lisa, anything to add?
No, I think both of those comments probably are adequate. I think in terms of our view, or maybe speaking for myself, what you allocate to H2 tilde in Frank's example is less important than the ability to control the family-wise or the study, I guess if these are all primary, I can't remember if they're primary or primary and secondary, but if they're all in the primary family, having the strong control would then let you say that you've demonstrated evidence of effectiveness with respect to the short-term symptom and the long-term symptom, and that, can factor into regulatory decisions and marketing decisions also. So I think our interest, the sponsor can decide how they want to divide and allocate, but we want to hold on to the ability to know, or the ability to quantify whether we're making a wrong conclusion.
Okay, thank you. So I think all the panelists, in fact, I think all the attendees can see the question that was asked. I think it's a fairly brief answer, so I'll ask the panelists if you want to comment on the explanatory endpoints. There are no requirements for type 1 error control, but the question is there some sort of control that might be expected? Any of the panelists?
Well, I'll jump in, although I'm probably the last person to answer this, or the least important voice here. So I think we actually downplay exploratory endpoints or their role in our guidance because we think they're not so much a concern for us. I think these endpoints, they might provide supportive information for us about how the drug is working or the mechanism of action, the duration, something else that might be important to understand what the drug is doing. But the fact that they're outside of our strong control space means that we're not considering anything to be demonstrated with respect to those endpoints. So we're not looking at efficacy being demonstrated with respect to those. So they're in the trial more, I think, for the sponsor to possibly explore the utility of that endpoint for another trial, perhaps. And so maybe from that angle, it'd be better to hear from Frank or Alex.
Yeah, Frank here. So I think I would agree with that. I see it primarily then for internal decision making, and as such, you certainly want to understand what are the consequences if I'm doing those exploratory analysis, and so you can do what makes sense, so to speak, but maybe some ways are more sensible than others. And if indeed you have a long list of exploratory endpoints, you might think about false discovery rates or some other sort of adjusting for multiplicity. It's really, what's the role of the exploratory analysis and endpoints in the overall internal decision process. That's how I would see that.
I think that's well said, Frank. I would say, just to say it maybe in a slightly different way, is that exploratory endpoints are hypothesis generators, and you make a very good point that it's up to the sponsors to be careful not to just look at 50 of those, and the one that looks best says, okay, we're going to invest in this phase 3 trial looking at that endpoint. That could be regression to the mean. You want to, for sponsor benefit, not have too high of a probability of a type 1 error. So let me, I hope, Alex, you don't mind. I'd like to move on. We have 9 minutes left. I want to ask one question that very interestingly, literally minutes before this meeting, I saw a pink sheet from yesterday. I'm just going to tell you the title of the pink sheet, which is FDA's Guideline Pushing Seamless Clinical Trials for Faster Development. And I haven't had time yet to read it carefully, but I believe that what he would be referring to is inferentially seamless. Operationally seamless is, to me, just semantics, having two studies in one protocol. At the end of the first phase 2 part, you do your analysis, you unblind everybody, and then you start the phase 3 after. So operationally seamless is really nothing that controversial. So for inferentially seamless, where you want to, at the end, to do an analysis of the combined phase 2 and the phase 3 part combined. And so my question to all of you are, if you have, for example, several doses being evaluated in the phase 2 portion, and maybe even some Bayesian longitudinal modeling, so maybe your primary endpoint is 6 months or a year out, and you want to be able to choose the doses that will then go into the phase 3 portion. I have experience of one of those kind of trials where simulation needed to be utilized to demonstrate, I'll put it this way, demonstrate some control of type 1 error. It certainly can't prove it, because the null space is multidimensional, at least in the example I have in my mind. So my question to the panelists is, do you think there's room for that sort of trial, and if so, where? I'll just offer that maybe it's the rare disease opportunity where patients are very limited, the number of patients in the very rare diseases, but I'll leave it at that and just ask the panelists if you have any comments.
So I'll jump in quickly. So if you're talking about a general seamless phase 2-3 trial where data from both phases are used or contribute to the analysis intended to support efficacy, establishing efficacy.
Right.
So that's a topic in our adaptive design guidance, not our multiple endpoints guidance. And in fact, that's one under the 21st Century Cures Act and the Prescription Drug User Fee Act number 6, to do the 6. And under both of those, we have a project and a commitment to generate a guidance about complex innovative designs, because I think we're a little bit discouraging about those designs in the 2010 adaptive design guidance. But in fact, we will accept those designs. The problem is you just have to show that you've got good control over the probability of making an erroneous conclusion, which I use instead of type 1 error in case you're doing something other than the frequentist analysis. And it may require simulations to show that. But I think that will be a topic of discussion under the public meetings and the future guidance that come out of the 21st Century Cures and PDUFA 6. And we'll have that first public meeting, I believe, in March of 2018. There'll be an announcement about that soon.
Okay, thank you, Lisa. Alex or Frank, anything to add?
This is Alex. Could just say very quickly that my experience with rare diseases is somewhat limited, but the general idea of applying a potentially slightly relaxed set of criteria to ensure alpha control in those cases seems quite reasonable to me. At least, we could consider using a simulation-based approach, something that would make us reasonably confident that the alpha is controlled, but perhaps we don't have to use a formal analytical framework, which simply may not be available.
Great, thank you. Frank, do you have anything, or can I go on to another question?
No, let's go on. Thank you. I think Lisa and Alex already provided good comments.
There's a good question in the chat box. Well, let me go to another question that I had prepared. And this is in the multiplicity guidance. The issue of safety endpoints, where there can be hundreds. The general thinking, I think, is no multiplicity adjustment of any kind. And there is a method that Joe Hayes and Devin Marotra have published on that use FDR for a large number of adverse events, for example. And do any of you have any comments on whether you think the FDA and their guidance should consider recommending some kind of control? And I think everyone would have to understand that the FDA always has the right and ability to say we cannot recommend approval, even though after you adjusted in the way you adjusted, it wasn't statistically significant. Those 5 deaths that were all definitely due to the drug is a reason that we're not going to approve it. Do you have any comments on that, or do you think we're going to just go forward with no multiplicity adjustment of any kind for safety endpoints?
This is Alex here. I can just quickly jump in and say that this is an issue that I would argue applies also to other types of exploratory analyses. I mentioned in my slides that I think the same general idea would apply to numerous exploratory subgroup assessments that are typically conducted in most phase 3 trials. I think it's a good idea to apply a meaningful adjustment that mostly with the ultimate goal of informing the decision-making process, because the problem here when we look at those multiple analysis of adverse events or multiple subgroup assessments, the ultimate goal is not clearly stated, and therefore it's not really clear how to formally define the type 1 error, that rate that we're trying to control. So anything that would facilitate the decision-making process, I think we should generally be welcome.
Yeah, and this is Lisa. I think that this is one of the issues I mentioned, that we got some comments on the public docket about the current guidance. What can we say about safety endpoints that are not in a family? And I think the difficulty here, I mean, we certainly run trials where safety is the primary endpoint. These might be a post-market commitment, or they could be a pre-market commitment to rule out the risk of some adverse event. And we do, worry about multiplicity there. But maybe more often, or what this question is about, is when the trial is run with efficacy endpoints and then you see something in the adverse event space and you have no plan to control for multiplicity. But that's the problem, right? That we have to react to whatever we see in the safety domain, and you don't often know in advance what you're going to see, and so it's tough to set any kind of spending function in advance. But because you also don't know how many you're going to be looking at. But that being said, I think that the statisticians certainly and clinicians inside FDA are aware of the chance of something spurious the more things you look at. And, when we look at safety data, we do try to take that into account. But you're right, it is informal at this point and not so much formal, unless safety is the primary endpoint.
Maybe just one more observation as we talk about safety and multiplicity adjustment. Probably safety is by itself a much larger problem, and it would be interesting. I think it's more important to think about a quantitative risk-benefit framework and having that one implemented properly. And while multiplicity adjustment is a nice technical aspect that is certainly also important, I tend to first think about how you actually think about, safety more holistically, so to speak. And I think there have been some, obviously some excellent work being done on this risk-benefit considerations. And I mentioned just this because sometimes even in our own companies, even this quantification of risk-benefit is not always given, so to speak. So just to add the perspective that safety obviously is a big problem generally, and how to tackle that and how to evaluate safety.
Okay, thank you, Frank, very much. I know we're at 11:01 right now, so I will take the opportunity to thank Dan for inviting me to moderate, and thanking all the speakers and panelists. Very interesting. We did a lot in 1 hour. So, Dan, I think I'll turn it back over to you.
Thank you, Walt. And again, I want to thank all the panelists, Lisa, Walt, Frank, Alex, and Ray. When you're starting out a new series, it's great to start off with a distinguished and all-star lineup. So thank you all for participating. The attendees, I hope you found it informative. I hope this type of forum is something that you can get good information from and enjoy. As I said, we're intending to do this quarterly, so we should be back again either in December or January for another one. I'd also like to thank Christy Trong-Stein, who really was the person who did most of the organization for this and getting the speakers, and Kara Hackman for the technical help. And with that, we'll sign off and thank everyone. And as I said, hope to see you next time.
Thank you.
Thank you. Bye-bye.
Thank you so much, everybody. Have a good day.